He has completed his Masters of Science in Biotechnology at University of Kerala in 2008. Further, Dr. Korean completed his PhD in Biotechnology at Cochin University of Science and Technology in 2016. Dr. Korean has completed his postdoctoral research at National Institute of Research in Reproductive Health (ICMR). He has more than 8 years of academic experience. Currently, he is working as assistant professor at BS Abdur Rahman Crescent Institute of Science and Technology in School of Life Sciences. Dr. Korean has received his CSTE postdoctoral fellowship in 2017. He has also been DST Inspire Junior and Senior Research Fellow from 2012 to 2016. He received first rank in Masters at University of Kerala. He has cleared CSER NET examination. Moreover, he has 26 publications with 1,700 citations with an H index of 12 in highly reputed journals. We are very much proud and we have so much pleasure to have you here, sir. Thank you very much for joining us today. And you can take over the session now, sir.
Thank you, and thanks to all management of Sri Shasta Institute of Engineering and Technology for inviting me to this lecture. So I am happy to hear that one of my friend is also presenting afternoon, Dr. Quincy Vijay Babu. I think she's one of my friend. She's also presenting. I think it will be also a very interesting session. So let me go to my presentation. Okay. Okay.
Thank you, sir. Thank you. Definitely.
This
Thank you.
Let me share. So hope you can see my presentation.
Yeah, it's visible, sir.
Yes, yes, yes. Thank you, man. Thank you. So for last 10, 12 years, I am working on melanin pigment, mainly bacterial melanin pigment. Nowadays, we have started the fungal melanin pigment also. So I am going to present my research. That's my research, and it has a lot of significance in the modern world. In the case of therapeutics, in the case of cosmetics, melanin has a great role to play, which I think that's why I'm sticking on with this for a long time. My topic for this webinar is microbial melanin and its application and insight. I am assistant professor at BS Abdur Rahman Crescent Institute of Science and Technology at the School of Life Sciences. So I'm happy to present my research in this webinar. So you people know that nature is full of colors, is it? So everywhere, every organism has a lot of different colors. You can, if it is a terrestrial organism or if it is aquatic organisms, everywhere we can see different colored organisms. Different world is full of colors, and we human beings also. You can see that the bottom picture. We also has different colors. We also have different colored in nature from what ranging from white to black, different shades of color we also have. If we go to what oceans, we can see different colored fishes. So colors makes our life colorful, is it? So can you imagine a laddu or jalebi without a color? Without color, whether we will, it will be appealing for us. So aesthetically, we one of other example is with our dresses without color. Can it is aesthetically good? No. So the world is full of color. We are in search for different colors, and so we are going to study about the colors, the colorful organisms in nature. Coming to bacteria also exist in different colors. I think you people maybe have attended many these agar art programs, making art with making pictures with these different colored microorganism in a petri plate. I think you have seen or you have done this. You can see bacteria as different colored bacteria exist in nature: yellow, green, blue, red, blood red, black, different colored organism present. With that organism nowadays, people are creating lot of pictures in the petri plate. So it is called agar art. People who don't know, I think people will be familiar with that. So people are creating a lot of pictures with this agar, sorry, this colored bacteria. So bacteria also exist in different colors. I think you people have done what pore plating or spread plating. After this, when you look, I hope you people are from biotechnology and done some micro work. So you people have done some what pore plating or spread plating or something. You can see a lot of colonies in the second day. Second day, you can see all these colonies are of different colors. Like that, bacteria also exist in different colors. So bacteria mainly these are the main colors produced by bacteria. Mainly one of the main color which is produced by bacteria is carotenoids. Carotenoids are one of the main color. They are yellow, yellow, green, red, different colors they produce. Then coming to phenacins. Phenacins is blue to green color. Mainly the pseudomonas type of bacteria produce phenacins. These phenacins are blue to green color in color. I think you people have grown some pseudomonas. If you grown some pseudomonas in the broth will appear like a bluish green in color. This is sort of the presence of phenacins, such as pyocyanin. So another pigment produced by pseudomonas. And prodigustin. Prodigustin is another pigment. You can see it's blood red in color. This color is a blood red in color. Prodigustin is a blood red colored pigment produced by bacteria. And violet colored pigments also produced by bacteria called violacin. So different colored pigments are secreted by the bacteria. And there are bacteria which secretes black colored pigment. You can see this broth seen in this bottle. This is just when I am inoculating this broth, it was white in color, but bacteria produce a black colored pigment, which is we melanin researchers consider as a black gold of bacteria. So that is the melanin. So I am going to tell more about melanin pigment. You know where melanin exists. Melanin exists throughout every organism. It's ubiquitous in nature. Most of the organisms have a melanin in some part of their body. So in humans, people all have melanin. Is it melanin main function? You know, but it is. If you don't know, I am saying it is UV protection in nature. Melanin help us protecting from UV light from the sun. So melanin protect us from UV light. So you people know that UV light can create thymine dimers. Is it? Like that, UV light can create a lot of mutation in our DNA, which can lead to cancer or like that diseases. So in order to prevent these mutations, we used to have a sunscreens. Is it when going out in the sun, we usually have a sunscreens. So melanin is found to be a what we can use it as a very good ingredient in sunscreen. Other than that, we can consider melanin as a natural sunscreen of our skin. We can like that we can say we apply sunscreen. Other than sunscreen, our natural sunscreen of cell, our body is our melanin, which protect from UV rays. So there are variation in the skin color from the in different from different ethnicity. The skin color differences can be seen, and the skin color differences mainly these sorry melanin is produced by the cells called melanocytes. Okay, melanocytes in the epidermis, and melanocytes secrete these melanin in the form of melanosomes. Melanosomes are the vesicle like secrete in the form of vesicle like structure. These melanosomes resemble everybody know about lysosomes. Is it? Lysosomes are contain enzymes which can which can kill the pathogenic melanosomes. It is full of packed with melanin. So this melanin is produced by melanocytes in the form of melanosome, and these melanosome after the production it will migrate to a specific type of cell called keratinocytes, where it will concentrate. We can see here in this picture, you can see the melanocytes down the epidermis, and it will secrete a lot of dots. We can see. So a lot of dots we can see. These dots are the these melanosomes. This melanin pigment is indicated as melanin pigment. That is the melanosomes. They are migrating to keratinocytes. You can see the keratinocytes. They the top, the keratinocytes. These keratinocytes, they will locate at the keratinocytes, and they will get concentrated there. So these melanins produced in humans, melanins produced by the melanocytes, they are transferred to the keratinocytes and localized there, and there dispersion to bacteria. Discuss how we get the skin color. So actually, this melanocytes precursor cell melanoblast is already produced during the embryonic development itself, which later developed into this this melanocytes. So these cells have a great importance in our body, and pigment also has. So if the melanocytes is lost, we can see the people with the patches of white patches in their skin called vitiligo. Vitiligo can occur. So melanocytes have that much importance in our body, and this pigment also have a. And presently this pigment has a very what it's so costly in nature. Melanin cost about $353 compared to gold, palladium. It's very costly in nature. And if we try to buy in Sigma, I think it's a sepia melanin. Sepia is the cuttlefish melanin. From the cuttlefish ink we can produce this melanin, and this melanin is cost about 9,000 rupees for 100 mg. So it is very costly. We are trying to produce more cheaper melanin with the cheaper media sources, etc. So many institutions are working on this. This is a melanin produced from cuttlefish by ICAR, Institute SIFT in Kochi. Like that, many institutes are working on melanin production and its utilization in various applications. We will see some applications later. So in the CSAR is working on, ICAR is working on, many institutes are working on this marine melanin sources. Melanin are still now itself melanin is being commercialized, but not much. That is our aim, is to make it more commercialized. We can see sunglasses with the melanin coated sunglasses. That is during the person come out of the sunlight, UV can harm their eyes. So in order to prevent the UV mediated damage, sunglasses are made with the melanin coating, as well as sunscreens are made with the melanin containing sunscreens. It can enhance us. Our study itself, we have found out that it can enhances the sun protection factor. Or I think you people have seen when we buy the cosmetic creams, SPF 40, SPF 70, like that. These are that, like that. Sunscreens. The SPF has a very important role, because the SPF denotes sun protection factor. Addition of melanin to the sunscreens can enhances the this sun protection. So this is what we are also working on this. And as I said, the melanin is distributed. We can be seen everywhere. We can see it in seed coat. Isn't where you have seen many black seeds of the plants. Many seed coats we can see melanin. It acts as cell wall centroids in plants. It is seen in skins of humans as well as various animals. We can see the melanin. Our hair we can see melanin. And what microbes, fungi, bacteria all produce melanin. Actinomycetes all produce this melanin in very good amount. Actually, Its main role is the protection from UV light, but we have found out that is melanin has a lot of other utilities or other roles also. We can see this the below picture. I don't have a cursor. The below picture you can see some cross mark. That is Chernobyl. Chernobyl. You have heard about Chernobyl nuclear power plant destruction. During the nuclear power plant destruction, none of the organisms, living organisms, can survive in that area because of the this nuclear radiation. But it was found that melanin producing fungi is inhabiting the down picture, inhabiting the areas where the nuclear radiation is very high. So it was found that this melanin is protecting it from what nuclear the protecting the organism from nuclear radiation. Like that, it's found to be radio protective, heavy metal protective. Lot of research shown that it has a lot of different protective well need to well study and actually the I have shown a structure of melanin in the middle, but actually the melanin is a amorphous compound which is we will come to that which is not soluble in nature. So yet the exact structure, these are the this structure is a part of melanin, that is structure is shown here, DHICA structure is shown here, but the exact structure of the polymer is also not at found out. So we were working on a lot to explore on this magic molecule. That is what we are focusing on this more. So this pigment is mainly produced from the amino acid tyrosine. You can see the top the tyrosine. The tyrosine metabolism lead to the production of melanin, lead to production of mainly in humans, and as well as the same melanin can be seen in bacteria, fungi, etc. And mainly via tyrosine via dopa. You can see dopa. Dopa via dopa, melanin is being synthesized. Melanin is called dopa melanin. Dopa, I think you have seen, you have heard about dopa. Dopa is a precursor of this dopamine involved in the neurotransmission. So this pathway only dopa is also synthesized. Via dopa, the melanin is being synthesized, and melanin is mostly seen in our Indian population, etc. Indian population. While if cysteine is added during the pathway, cysteine, you know that cysteine is the sulfur-containing amino acid. If cysteine is added to this during this pathway, if cysteine is added, it can lead to the production of pheomelanin. Pheomelanin is a red melanin. Melanin is a black-colored melanin. Pheomelanin is a red-colored melanin, which is mainly seen in this European Caucasian people. That is, people have this pheomelanin abundantly in their body, and mainly come to there are several others melanin, other melanin also is there in plants. The melanin is synthesized by a catechol. So in fungi, you can see the melanin is synthesized is called DHN melanin, dihydronaphthalene melanin. So the predominant melanin in fungi. Coming to bacteria, predominant melanin mainly pseudomonas, pseudoultramonas, this type of organism synthesized by a homogeneticity. That type of melanin is called phyomelanin. These two melanins are black in color, but they differ in their chemical nature compared to the melanin, but their physicochemical properties are somewhat similar. So we call all these as melanin. And while I'm saying about the structure, actually the problem is of melanin is insoluble in water, and it's soluble only in alkali. And this is the work we have done. It is insoluble in water. This makes it difficult to determine its structure. That is the solubility in different solvents is being displayed because it's insoluble in most of the organic solvent. Using it for different applications is it's a major limitation here. So that is the main problem regarding the melanin. So basically during these years I was focusing on bacterial melanin, and why I chooses bacterial melanin is is that bacteria, mainly this actinomycetes bacteria. When we come to actinomycetes, streptomyces is the bacteria which produce a very good amount of melanin, and they produce melanin in the natural environment. But rest of the bacteria will not produce the melanin in their natural environment. We are trying to induce it for the production of melanin. That is what we are doing here. Bacteria normally, humans produce melanin naturally, as well as fungus such as aspergillus. If you see aspergillus, you can see the colony become black in color. So they produce melanin naturally. But bacteria, most of the bacteria won't produce melanin naturally. So we are trying to produce melanin by induction of melanin by giving special medias. So why we are doing this induction of bacterial melanin is that when we are trying to extract melanin from our hair, is it, we have a lot of hair waste will be coming, so we can extract the melanin from our hair, and or take protein contamination will be very high. I mean, our hair contain keratin, like proteins are there. So in order to separate from these proteins, melanin from these proteins, it is too costly thing. So while we are using bacteria, the advantage of bacteria, even if it is fungus, also fungus secrete melanin in the inside their cell. But bacteria secrete melanin to the media. It's extracellular. Secretary. There is any product produced in one batch will be differ from other batch. So batch variations will not be there in the case of bacterial melanin. These two things make us focus more on bacterial melanin. But still, the problem is that getting melanin from hair, melanin from sepia ink, we can get a lot amount of melanin. Bacteria produce that will not produce that much amount of melanin. So very not that much melanin is produced with bacteria. This much mg or 1.5 gram per liter. This is about the melanin production by different bacteria, or the maximum of 6 gram per liter, 21 gram per liter is maximum. Is it? Yes, 21 gram per liter by streptomyces. Streptomyces produce 21 gram per liter. So different bacteria produce this much only the pigment we can produce. Using bacteria. So our main aim is to try to enhance the melanin production by using media optimization, mutation studies, and creating genetically engineered organisms. All these are going on globally, as well as we are also trying to do that. And this is one of our bacteria. It is produced about 1297 microgram per ml after optimization, pseudo ultramono. So you can see, I just taken this PPT to see the media. The first white media is the initial media. After 5 to 6 days, the media will turn into this black in color. So that is the very thick black colored melanin will be obtained, and we can remove the bacteria, and we can just extract the melanin very easily. And coming to melanin's functions, as well as its applications. We can see melanin already tested in solar cells as a what are this what a sunlight sequestering compound, as well as it is used as an anodic compound in batteries, optical lenses, which we have discussed, cosmetics, and it is used in vitiligo. I think vitiligo people, you have seen the white pattern in the skin like people. These people, vitiligo, there is no such treatment people have heard different places treatment are there, but actually total cure is not possible, because actually medically it is the melanocytes have been destroyed. So it is and how it is happening is also still hypothesis. It is already accepted hypothesis is it's an autoimmune disease, that our autoimmune system is destroying these melanocytes, and several other hypotheses also still exist. So we need to we are still exploring this pigment associated disease also. So only solution is we can have some creams which can mask this, so that that usually these people who have these white patches where in face or somewhere, they will be having through some psychological problems. So that psychological problems can be minimized by using melanin containing vitiligo masking creams. And this also found as it has anti-venom property, photoprotective property, thermoregulation. It is involved in nitrogen fixation. It is involved in electron transport pathways in different organisms. It also found as it has anti-venom property, photoprotective property, thermoregulation. It is involved in nitrogen fixation. It is involved in electron transport pathways in different organisms. It acts as a pathogens our immune system and infect us. So, a lot of implications are there. Melanin associated with virulence, virulence associated with this nitrogen fixation. A lot of implications are there. This one of our study found out that this melanin secreted is a TEM image. Melanin secreted by the bacteria. That is also we are in a confusion whether the melanin is secreted by the bacteria or in the media melanin is being formed. That is also we are trying to explore. A lot of things, a lot of gaps are there everywhere in melanin research. So here we found out that in media, you can say like that. In media, the melanin appears as a small particles, small ultra nanoparticles, about 10 nanometer to 100 nanometer, ultra nanoparticles, which is of high stability and which is stable up to 1 month. And as it is this much stable, we could use it as a drug delivery device. Many people have been researched so far and find out it could be used. We have not done it. We are planning to do it. Is using as a drug delivery device because of this very high stability and very small nanoparticle in size. And it was found out that it not by us, but it was found out that in melanin can even cross the blood-brain barrier, and it has neuroprotective nature. A lot of studies has been done so far. And melanin, our study found out that melanin has a very good anti-inflammatory activity in vitro conditions in raw cells. It was found out that our melanin is inhibiting the what these inflammatory enzymes, cyclooxygenase, lipoxygenase, myeloperoxidase, etc. So this melanin is been having anti-inflammatory activity. Later, our what collaborating group have found out that melanin is also this inflammatory activity. Melanin is showing some inflammatory activity. And we have found out that melanin has anti-cancer activity against these lung carcinoma cell lines. Melanin shows anti-cancer activity, and it has shown destroying the cancer cell. Our apoptotic staining shows that melanin is inducing apoptosis and killing the cells, not by necrotic pathway. And it is showing at a very low concentration. It is showing very good anti-cancer activity. This also researchers has proved in later it is proved in vivo condition that is as anti-cancer activity, and more research need to be, or more trials need to be done so that it can be used yeah, and further. Most of the many result will come like that anti-cancer activity, but we need to work on more on this. We can't say that it can't be directly used as a medicine, but we need to go for a lot of trials to confirm that it's melanin's activity as anti-cancer compound. And I think you people have done this antibiofilm or antimicrobial assay. I think you people have already done this antimicrobial antibiotic susceptibility disc assays, etc. So this antimicrobial our picture is not from our work; it is some others' work. It was found out that this melanin pigment was found to be antimicrobial in nature. It can inhibit the growth of bacteria, and this antibiofilm work was done by us already. It was found to inhibit the biofilm formation by the very good biofilm forming bacteria. It was found to inhibit the biofilm formation. You can see it in the same picture also. The control is seen in first, and the second is the treated one. There is a lot of or the killing of the or the biofilm is get disturbed in the second picture. You can see we can't tell that killing of the cells maybe biofilm, the extracellular polymeric substance is being destroyed during this, so that they have just so the biofilm formation is being disturbed here by melanin pigment. And antiviral activity, melanin has found to be having a hiv anti-hiv activity, and different viruses has been infected, I am sorry, inhibited by this melanin pigment. And there is a in silico study which found that melanin precursor is found to could be a drug which can prevent the binding of spike to the lung epithelial cells. This we need to work in lab, then only we can confirm more on this. So it had shown to have some antiviral activity. We will be working on this also. Antiviral activity also. We are working on one thing like this. This is all about research which we have done, and many people have done more radio protective activity. It has property of removing heavy metals from water, so that it can be used for water purification. Lot of research has been doing in melanin, and many premier institutes in India and many companies also focused in melanin based products. So I am also trying to develop a product or something from this, and hope this presentation help you also to explore more. And the pigment is a pigment which is less explored. You can explore more in the pigment. We use different techniques for the characterization of melanin. We use use spectroscopy, and mainly this pigment absorbs well in UV region. So it gives a characteristic peak in the UV region, and FTIR, SEM, TIM, as well as TGA can be used to determine heat stability, as well as ESR spectrum can be used to determine its radical scavenging activity, because it is a very good antioxidant in nature. And X-ray crystallography can be used to find out its amorphous nature. Usually this melanin is amorphous in nature, and several other NMR all are used to determine the structural properties of this pigment. And this pigment has a lot of things to explore more, and we are working on that. Hope you got some idea about the melanin from my talk. Thank you.
Thank you very much, Dr. Korean. Now let's move on to
Yes, ma'am.
the question and answer session. Students, do you have any queries?
Yes, yes.
You can interact with sir. Do you have any queries?
Something in chat. Yes. No, no, no. Sorry. If you have any queries, you can ask. Speak, man.
You can interact with sir if you have any queries. Or you can post your questions in the chat box.
Yes, yes, yes. Some questions. it has found to have what's it? Kushi. It has found to have anti-venom activity, but it was in preliminary studies found that it has anti-venom activity. It is not a peptide. It is a phenolic. It is actually melanin. I have not introduced that. Melanin is not a protein compound or peptide. It is a phenolic compound. It's a phenolic chemical, just like lignin or like that, phenolic chemical. So we need more to study its anti-venom properties. It was reported that it has some anti-venom activity, but we need to what go into more studies. Then only we can be used as a anti-venom medicine. It is not a peptide in nature. It is a just a phenolic compound. Okay, thank
Any
you.
other questions? Any other questions, students? You can post your questions in chat box, or you can interact with the speaker.
Yes.
Uh, I now request Mr. Gomati Shankar to propose the vote of thanks. And before that, the feedback form has been posted in the chat box. So the certificates will be given for the students who are filling the feedback form. Thank you very much. The certificates will be issued in 2 to 3 days to all the participants who have filled the feedback form. Now I request Mr. Gomati Shankar to convey the vote of thanks.
Thank you, ma'am. Good morning, everyone. On behalf of Department of Biotechnology, I extend my heartfelt gratitude to each and every one of you for joining us today for our webinar on microbial melanins and its applications. A special thanks to our esteemed speaker, Dr. Nobel Quirian, for delivering such an insightful presentation. Your expertise and passion for the subject have truly enriched our understanding and inspired us to delve deeper into this fascinating field. To all our participants, thank you for your active participation and engaging questions. Your enthusiasm and curiosity are what make events like this truly meaningful. I would also like to thank our HOD, Dr. Sai Preeti, and the organizing team for their hard work and dedication in putting together this informative session. As we conclude today's webinar, let us carry forward the insights that we gained today. Thank you once again, and we look forward to welcoming you to future events as well. Have a wonderful day ahead. Thank you, sir.
Thank you, sir. Thank you, ma'am, for this opportunity. Thank you.
Thank you very much, Doctor. Thank you very much.
Can I leave?
Yeah, yeah, sure, sure.
Okay, thank you.